Good morning and welcome to the pan-genome consortium webinar. My name is David Newman, Territory Manager for Energene. In this webinar, we will go over why is the pan-genome needed, the implication it has in cotton breeding, and the status of the consortium. Joining me are Dr. Roy Cantrell, former global cotton breeding lead at Monsanto, and Dr. Paul Comet, technical expert at Energene. We will dedicate the end of the session to live discussion, and we encourage you to participate. You can use the question tab on your attendee dashboard and either write us that you want to ask a question, and we will open your microphone and give you the option to speak, or you could write your question and we will answer it. Roy, the stage is yours.
Thanks, David. Good morning and good evening. Thanks for joining this webinar. I started working with cotton DNA markers in 1990 when I set up an academic lab at that time. I have become involved with Energene because the technology addressed here addresses many of the challenges and frustrations we faced down through the years. Most of the time, genomic or genetic variation is detected through the use of a single reference plant genome, which is assembled from a single plant variety, which in cotton may be TM1 or some other plant variety. Low-level resequencing data is now readily available, as depicted here. You can get reseq data from diverse germplasm and map it back and compare it to the reference genome. As you do that, ideally, as is shown here, it should align with the reference genome. However, this is not always the reality, as is demonstrated here. What usually happens is you have reads like is shown in blue that aren't mapped. You have reads like the ones in red from reseq data that stack up, and you also have gaps. This leads to a high rate of false discovery of polymorphism due to misalignment, a high rate of undetected polymorphism, as shown there in blue. You have, or in the gaps, you're not detecting polymorphism, and you have limited discovery of only a part of the polymorphism. You're not able to detect all of the SNPs or all of the structural variation. On this slide, this demonstrates just a figure showing 3 genomes that can be aligned into a hypothetical plant genome, that pangenome, that is able to analyze and detect structural variation. Energene provides a holistic approach here. This is able to use transcript analysis to locate transcript area, match the transcripts to their genome location, and through annotation detect presence, absence variation, as well as copy number variation, indels, and translocation. This allows transcript analysis and annotation to couple it with the accurate genetic mapping information. The immediate and future utility of a pangenome is shown on this slide. In the short term, a pangenome of up to 5 or 6 Barbadensia and Hirsutum genomes allows a very cost-effective multiple reference genome. You access full sequence comparisons. As I stated before, structural variation is detected in a way that's never been available for cotton, and you have down to gene level allele sequence identification and sequence positioning across genomes, as is shown there at the bottom. This allows future capabilities which enables improved arrays, genotyping arrays. It's very important to note many researchers in the public sector and the private sector have existing genomic data, possibly from SNP arrays or whatever, That data is not lost. It can be incorporated into the haplotype and genomic data that comes from the pangenome. Once the haplotype and pangenome data is available, then genotyping using high-density sequencing becomes a very affordable way to detect markers in a molecular breeding program. The data is structured in a way that imputations are is involved in markers through the genotyping process. And this streamlines marker discovery, association with traits, and haplotype-based mapping and allele discovery becomes more readily available than it is today. We don't have time to go into all of the post-pan-genome solutions of NRGene, but they're shown on this slide. Once the pan-genome is completed and the databases are created, you go beyond the physical reference genome at the bottom to comparisons across your diversity that exists within your breeding program, the haplotype diversity that exists within your breeding program. The diversity analysis enables better analysis of your germplasm as well as marker discovery. I mentioned that this gets breeding programs into a place where genotyping through high throughput sequencing at a very low coverage after the pangenome is created really makes affordable genotyping by sequencing a reality in cotton. We all want to get to a place where we can do predictive genomics, genotype analysis, QTLs, GWAS, genotypic selection, genomic selection, and molecular breeding platforms. All of these are post-genome technology solutions post creation of the pangenome. If we talk about cotton, what are the potential implications of this? If you've worked in cotton before, you probably know that genomic variation in the primary gene pool of Hirsutum and barbadense is likely modest. I've worked in wheat, soybeans, other crops, and for a self-pollinated crop, you have modest genomic variation detected by SNP markers or SSRs or whatever. We've always faced that in our molecular breeding program. That motivates me, and it's imperative based on that experience that all genomic variation, both SNPs and all structural variation, be detected and explored in the primary gene pool. Detecting this genomic variation enables targeted integration in key genomic regions. I refer to this as precision pre-breeding. If you look across the genome, you can identify using the integrated genome viewer a very specific segment of a chromosome in your germplasm to increase haplotype diversity. I refer to this as precision pre-breeding. And this is a more precise way of dealing with genetic diversity and integration in cotton than we've been able to do before. My experience in cotton is pre-breeding is complicated, challenging, because you're introducing a lot of diversity and not targeted to particular genomic regions. And I think we're approaching a time where we can do targeted integration. I think the most exciting thing to me is the ability to detect structural variation. This can unlock some mysteries in cotton from my perspective. My 30+ years of working in cotton, in some genetics backgrounds and some germplasm, approaching homozygosity from selfing or backcrossing may be slower than is predicted by theory. This has always been a mystery, or is recombination and structural variation in certain genomic regions a factor here? In programs where you use marker-assisted backcrossing to recover the recurrent parent, any program of scale has always experienced, regardless of the traits being the target traits, there's always certain recurrent parents or germplasms where recovery of, just like the prior point, the recovery of the recurrent parent is not what is expected based on number of backcrosses. This could be due to structural variation. I've spent years looking at hirsutum by barbadense crosses. It's well documented in the literature and from research experience that segregation distortion occurs in interspecific fertile populations between hirsutum and barbadense. Integration of traits between those two species has been hindered by that. Structural variation has been implicated as one of the factors in that, and I believe that being able to understand from the pangenome and the haplotypes created from that, we'll be able to track, understand in all parent combinations here the structural variations that are segregating and contributing to this. Unique to India cotton, where hybrids, hirsutum by hirsutum hybrids are common, heterotic group development is taking place in hybrid cotton and hirsutum in India, somewhat like maize. I believe understanding structural variation that exists between the development of heterotic pools could help better create smarter heterotic pools as they're developed. In addition to what I've had here on the slide, in some crop species, structural variation, small indels, other structural variation has been shown to have significant association with phenotypic traits or QTLs. As you do a GWAS, look at all the variation, SNPs and indels and structural variation, I think there's in some crop species a disproportionate amount of structural variation in some of those crop species that's associated with QTLs. So those are not detected on any of our major SNP arrays today or any of the variation that we're doing in GWAS or QTL studies. The haplotype data that's generated from the pangenome really helps us resolve the bias that exists in existing SNP arrays in cotton. We've all used the very useful SNP chips and arrays that are available in the public and private sector. Those have, the SNPs have some bias due to the diversity samples that were used to create them and the diversity that's sampled by the SNPs. Adding upon that, the structural variation and additional SNPs from the pangenome, I think strengthens our genomic knowledge in cotton. We've always, in my experience, faced situations where you identify QTL, you identify important genomic regions in cotton using SNP arrays, and almost invariably, if you want to use it in elite germplasm or in your, what I call breeding galaxy, a pool of parents, you have to find map that trait in order to be able to find polymorphisms across your entire breeding germplasm. So we need a combination of tools plus of using existing SNP data, which is not lost. You still use that, but you superimpose upon that all of the data created by the pangenome. If we look at the approach that Energene is taking, launching an ambitious cotton pan-genome project. To date, this is the status of where the pan-genome is headed, utilizing one public Gossypium hirsutum TM1, 2 germplasms from China, 1 barbadensia and 1 hirsutum, 2 hirsutums, and then we have 2 genome projects that are yet to be formed, but are very close to being finalized, 1 hirsutum and 1 barbadensia. So I think you see there you have the potential of 5 to 6 genomes in place, which based on an all-to-all comparison will create a virtual pangenome that really is powerful in the cotton community. The purpose of this webinar is to really introduce the consortium concept to the cotton community. On this webinar, solicit additional participants in the pangenome consortium globally. David, I'm going to turn it over to David, who will explain the consortium structure and where things are headed. Okay, David?
Thank you, Roy. Yeah, for sure. I'll go a little bit about the consortium structure and how it works. So to join the pangenome, each participant contributes an accession of their choice. The cost for an accession to do a de novo reference level assembly of the pseudochromosomes is $49,000. This is a very special offer for the consortium, as $49,000 normally covers for the assembly up to scaffolds and then the build up to pseudochromosomes. Energene is giving the pangenome comparisons and its deliverables free of cost in kind for all participants of the consortium, but that's only while the consortium lasts. As an exclusive offer as well, the first 2 participants to join the consortium after this call will receive $10,000 in credit for Energene's future solutions in other projects. The deliverables of this project include scaffold level assembly per sample, the sequencing and the analysis, the chromosome construction based on available reference genome, mapping all participant genomes. Each pair of genomes will have a map, the structural variation analysis, presence absence, copy number variation, the access to Energene's IGV viewer, which allows you to visualize multiple genomes at the same time, will be available for download. And consortium project will be starting on November 15th when we have received at least 5 genomes. And have you seen from the last slide presented by Roy, we are almost there. So we are getting there. Okay, so that would be on this slide, Roy, may you kindly, thank you. So I'm just gonna give a little summary as a recap. It's a quick recap. The pangenome offers a new method to capture sequence-based diversity in cotton through multiple reference level assemblies. The deadline to join the consortium is November 15, 2019. The first 2 customers following this webinar will get $10,000 in credit for future projects with Energene Solutions. For additional information, please visit our website at www.energene.com or contact an Energene rep. I will now open the floor to questions to the Q&A session. Thank you. If you have any questions, please send it our way and we will be happy to answer them. Okay, just received one. The question is, I don't have enough funds to contribute for a single genome. Would it be possible to work together with other researchers to fund the genome together? So yes, this is definitely possible. We have success in this strategy with other species in which we've had consortiums. So we do encourage this as well. Okay. Another question is, how much structural variation can we expect from the pangenome? Roy, can you take this question, please?
Yeah. I mean, I obviously have a keen interest in the amount of structural variation that's detected in the primary gene pool of Hirsutum and Barbadense. Uh, NRGene has conducted a pilot pan-genome utilizing 3 genomes uh, to really test this and to get the pan-genome algorithms and what's going to be used for the complete pan-genome going. That pilot pan-genome involved 2 Hirsutums and 1 Barbadense. Uh, and when you compared Hirsutum and Hirsutum, about the amount of variation that was structural variation, indels, other types of structural variation ranged that was in the ballpark of 15%. When looking at chromosome 1, they're expanding us to look at other chromosomes, but if you just take a sample of chromosome 1 in cotton and do a pangenome analysis of those 3 genomes, what's the level of structural variation? Is about 15%, which again tells me that's the variation we're not detecting with existing SNP technology. You compare Hirsutum to Barbadense, it's in the ballpark of 19 to 20% chromosome 1. This is a very preliminary analysis, but it gives you the ballpark of the amount of variation that exists potentially in cotton. For Hirsutum to Hirsutum, when you look at all possible variants or polymorphisms, it's about 2 for that same chromosome. It's about 2 polymorphisms per kb in a Hirsutum by Hirsutum match for chromosome 1. Obviously, when you compare Hirsutum and Barbadense for chromosome 1, it's over 5. It's about 5 times that number, about 5 polymorphisms per kb. As I said, this is a preliminary and pilot study, but I think it gives you a glimpse of the variation that can be detected. The level of polymorphism will likely change as the full pangenome is developed for all chromosomes. And I think it's important to re-emphasize that the full cotton pangenome is going to incorporate both Hirsutum and Barbadense. So I think it's going to be a very robust tool for looking at all the variation that exists in Hirsutum and Barbadense. So hope that answers your question there.
Thank you, Roy. We have another question. What technology are you using to do the sequencing? Saul, may you kindly respond to this?
Sure. So the sequencing is based on a number of different preset Illumina libraries, and these libraries go from paired end, PCR-free, all the way up to we incorporate the 10x genomics library preparation, which allows us to get very large segments, pseudo sequences, due to the 10x genomics capability of putting together a large, almost 100 kb piece of DNA based on the single droplet technology. So it's all based on Illumina, and that's not to say we couldn't. We do offer adding in PacBio sequence, and we use that for scaffold linking as opposed to the primary assembly. But our technology has always been and continues to be based on Illumina-based libraries.
Thanks, Paul. In the same wheelhouse, we have another question. What is the coverage of the genome covered during sequencing?
Yeah, so we have actually 2 methodologies, 2 analytic pipelines. What we call de novo 3. This is the 3rd version of our de novo assembly, de novo magic assembly. We're covering, total coverage with all the libraries is approximately 200-fold, so 200x. We've introduced a different assembly pipeline that uses fewer libraries, and coverage is about 95 to 100x on that, also using Illumina. So we're, depending on which technology we would apply, it's somewhere between 100 and 200x. So this gives us extremely accurate sequence information, since Illumina has high accuracy and we're covering the genome so high. Our best reference genome pipeline is the de novo assembly 3.0. So that's a 200x coverage, including the 10x genomics or Chromium library type.
So we have more questions, and we encourage an open discussion here. So I see a question from Pawan Kara, and Pawan, I will open your mic now, so you can ask the question yourself. If it's okay.
Yeah. Hi, thank you very much for this presentation. And I would like to know whether all the partners will be having this access to raw data, or only the one who has contributed will be, getting the raw data.
Yeah, so I can take that. So the partners in the consortium will share all the data. So both the raw data, the assemblies, and the files necessary for the pan-genome comparison.
You ask next.
Thank you.
Part of the agreement is to share this data.
One general question to Narjeen, that whether this is for only cotton, or is it for tomato or other pan-genome consortiums as well?
I can take that question. So we have a consortium already started for tomato. We are developing the consortium right now for cotton. We have also one for canola. We are developing for different species. So if you're interested in a different species, please contact me separately via email, or you can call me, and I'll be happy to give you more information about the other consortia that we are running.
Thank you. And also a follow-up question, that what kind of a support in the GS Narjeen offers?
Paul, please.
So support for genome selection, is that what you're asking?
Yes.
Yeah, so we do not have an algorithm building. We are looking in the future to support this. So our support for genome selection is really on the genotyping side, providing quality genotype data, haplotype information for a customer to use that data to build their genome selection models or applications towards selection, genome selection. So yeah, so we have tools. So we have a genomics tools software to get at the phenotype diversity information, but that does not include, like I said, preset or building of the genome selection models.
Mm-hmm. Thank you.
Okay, we have another question from Sachin Nikam. Sachin, the stage is yours.
Hello. Hello.
Hello. Yes.
So how much time it will be required to get, complete this project?
Yes. So once we collect all the DNA samples and everything looks all right, it takes about 6 months to complete the whole project. So deliver all the deliverables in the pan-genome.
Okay. Thank you.
Are there any more questions? We're happy to answer. I see a question here from PJ Kulkarni about yielding codon hybrids compared to current techniques. Paul, can you shed some light on it?
So I'm not sure of the question.
Paul, I think I can address that. If I understand, okay. Maybe he can rephrase the question. He's trying to see what the variation in maize is, hybrids between the heterotic pools versus what you may see in cotton that would help you design heterotic pools. Is that interpreting the question correct online? Am I understanding the question, PJ?
Yes, Roy, you are correct. Just want to have comparison.
Oh, okay. Well, I mean, there's been extensive work in maize identifying the large amount of structural variation that exists in maize, and to some extent that is helpful in understanding heterosis and the heterotic pools. And I think in a crop like hybrid cotton for India, PJ, it would be helpful as these heterotic pools are being created in cotton to understand not just SNP variation, polymorphism from SNPs, but understand all the structural variation that may help you guide the construction and development of the different heterotic pools. I think when you just, my experience in hybrid cotton, when you just look at SNP polymorphisms and genetic distance from SNPs, it's not always the best resource for diversity that relates to heterotic pools or hybrid vigor in cotton. We need more information, and I think adding structural diversity and haplotypes derived from that would be helpful. That's just my speculation. I don't have any data, but that's more speculation, PJ.
Thank you. Thank you very much.
Okay, another one from Sachinika.
Yeah, actually, I'm more interested to have utilization of this data in hybrid breeding. So how this data will be helpful for me to select out the parents for the better hybrids?
Yeah, I can maybe address that. I mean, what we've shown here with the germplasm going into the pangenome is either from China varietal programs or US varietal programs. So I think we need to have a discussion as to how and if what the pangenome construction would look like for India cotton. So I think we would just need to have a follow-up discussion, because when you create a pangenome, you want it to be based on the genomic diversity that exists within germplasm that you would use in your hybrid program. So just using what's in a varietal program may not meet that goal. So I think I would encourage anybody in India to have further discussions with NRGene and us about what would the pangenome and NRGene technology be applied to India and hybrid cotton. We need to have that discussion. And I'm not saying that the existing consortium cannot be expanded. I just think we need to have that discussion. I don't know, Paul, if you have, I know there's people from India on the call asking about.
One more question. Actually, whatever the SNPs data or the copy number variations data will be generated, whether we are going to have the chip of this database so that whenever we want to do genotyping for another samples, so it will be possible for us.
Yeah, so Paul, you might talk about how you're able to select and generate markers for any variation. Do you want to go over that quickly, Paul?
Yeah, so the pangenome is obviously constructed from these 6, will be constructed from 5 or 6 lines. Genetic variation in a germplasm base is obviously a lot more than that. Now, Energene's technology builds on the pangenome. So we take further sequence information and organize it in a way that allows us to also see structural variation and also position it relative to this pangenome. Once we've constructed that database, which we have constructed in cotton, we have, like Roy said, we have a pangenome based on 3 genomes, and we've incorporated, I think it's about 16 different diverse lines. But again, these diverse lines come from mostly North American breeding material. So with that database, then we can start designing the optimal set of markers that could be used for genotyping in basically any technology. It's sequence based. So we identify the markers. We have the ability to identify markers, sequences that can be converted to genotyping by sequencing markers or SNP-based markers, whatever's needed. But it's identifying that diversity, not being biased by a single reference, but using this pangenome or the pangenome of choice to get at the optimal variation or the most variation you can identify, because we're getting away from that ascertainment bias that occurs with a single genome. I hope that answers the question.
Yeah, thank you.
Thank you, Paul. With this, that was our last question during the Q&A session. I would like to thank everybody for participating. Please don't hesitate contacting us at Energene if you have any additional questions regarding the pangenome or other solutions that we provide. We'll be happy to answer them. We'll be looking forward to the start of this exciting project. Thank you once more.